Welcome back. We have this is the start of the afternoon session. Sorry if you couldn't finalize the lunch, but will be a break after the next 3 talks. Could have wished to give you a little bit more time, but we're a little bit on the time schedule because this talk not, the next talk going to be live. Then there's a Zoom, and the Zoom is of course going to be on time, sharp. So, but now it's my pleasure to introduce Patricia van der Wallen. Some of you might know it. She's from the MOVANT, so from the, correct me if wrong, from the department that studies movement. Well, you all know physiotherapists. My mother was actually a physiotherapist, and the people came in and she would, say, oh, this is not good. She would work on the muscles, and that was it. But times have changed, of course, and she's going to illustrate what that can hopefully mean for you and your children. Please go ahead.
Thank you. Thank you. So no introduction needed anymore. I'm going to talk about gait because I'm a physiotherapist, but I'm specialized in kinesiology. So we're studying the movement, and especially gait, because gait is a movement that is very cyclic and should be done each time at the same, each step should be the same. So it's very easy to study movement if we're looking at gait, because we know that in a normal gait, every step should be the same. I'm going to talk about the difficulties. I'm going to talk about how we can evaluate gait. And I'm also going to talk about treatment options for gait deviations. I'm trying to do it very specific for children with the ADMP syndrome, and I learned that I can use both terms, HEVDAS and ADMP, because I was really confused what was the right name. So I'm happy that I could listen in to the talks this morning, that I can use both names. But we don't have a lot of experience on this specific syndrome yet. So I'm going to talk to you about some specific patients that already did a gait analysis. Thank you to all the parents that gave permission to show the images and the data. I'm going to try to link it to other syndromes that we know with also intellectual disabilities and what we can learn from them, what we already learned, but what we hope to learn in the future also. So it's an ongoing topic. It's maybe new, but we do this now for about 20 years. Gait analysis, instrumented gait analysis, so not just looking, but really in-depth analysis of gait. We do it now in children with other pathologies, mainly neurological pathologies, such as cerebral palsy, and there we do it already for about 20 years. And we try to take that knowledge to a first pathology of intellectual disabilities. That's the Dravet syndrome, and we just finished a large research project, and we have a lot of expertise of the Dravet syndrome, and we're now trying to combine that with other pathologies to expand it and to help more children. So let's talk about the difficulties first. If you want to know what is a normal gait, then we can talk about prerequisites for normal gait. There are 5 prerequisites. If they are not met, then we can talk about the gait pathology. The first prerequisite is that I'm able to position my foot at initial contact. So the initial contact, and I hate these desks because I cannot walk on them, but the initial contact is when I put the first contact I make with my foot. This contact defines how I absorb my body weight and how the force, the ground reaction force, is working in the other joints, in the foot, in the knee, in the hip. If it goes wrong there, then it goes wrong along the rest of the gait phase. The second prerequisite is stability in stance. If I want to make progression during walking, I want to swing my leg, I want to progress, which is the goal of walking, then I need a stable stance phase. So I need to be stable on this leg while I can swing my other leg forward. If I would ask you to follow this presentation standing, not just on both legs, but on one leg and with a little bit flexion, then you understand stability in stance after 30 minutes. Because this is really the difficult part. If it goes wrong there, again, I have a pathological gait. I talked about the goal of walking. The goal of walking is progression. So I want an adequate step length, because if I take small steps, I'm not progressing. And that's the goal of walking. The fourth parameter is foot clearance. If I want to walk, I want to progress, I don't want to fall or trip. So if I'm not able to clear my foot high enough from the ground, then my toes might touch the ground and I might fall, trip, or even fall. And that's even more difficult when I go outside and when the floor is now it's wet, or when there are cobblestones, as you see them in Belgium a lot. Then you have a problem. Again, one of the prerequisites is not met, and we have a pathological gait. There are 4 that I can show during the gait cycle. The 5th one is energy conservation. That's something I cannot show, but the normal gait should be efficient. That means when one of the 4 first parameter goes wrong, I have to put a lot of energy in walking. I will be tired, I cannot walk long distances. Again, then we talk about a pathological gait. Gait difficulties in ADMP syndrome. I'm going to talk again about those prerequisites. I was talking about pre-positioning. In a normal situation, I have my heel that contacts the ground, but here we have 2 patients that we already tested in the gait laboratory, and we can see here that there is a problem with the foot, but also with the knee, which is not extended, and the hip flexion is not enough. This is another girl with the same syndrome, but here we see that there is a nice heel contact. The knee can be extended a little bit more, and also in the hip it might be a little bit more. The trunk might be a little bit more upright. But she has less problems than the boy in this picture. We tested already 7 patients. I tried to take the extremes, but we're not so far that we did a group analysis. So I can show you the pictures of gait that we observe in the syndrome, but I cannot tell you that this is a typical syndrome. We're not there yet. We hope that in a few years we can put all the data together and talk about what is typical or what gait deviations are typical for the syndrome. Stability in stance. If you stand on your foot, on the full foot, not on your toes, and it's difficult for those who are walking on high heels, they know what I'm talking about stability in stance. So a good foot contact is essential, but also the knee should be extended and the trunk should be over the body weight. This is all problems with stability in stance. More knee flexion, the trunk which is not over the body weight. Those are all problems that we can observe in ADNP. Adequate step length. If hip extension is not enough, then the step becomes smaller. This is the green line is what it should be. But also if hip flexion is less, then the step becomes smaller. Again, we observe children with smaller steps, wider steps also when you look from the front, but it's not the case in all the children. We also see children where there's no deviation from normal. The same for foot clearance. If the foot is not in dorsiflexion, so if the foot cannot be neutral, if you cannot flex the foot enough, then you might drag your toes. If that's the case, you can always compensate in the knee by more flexion. But if the compensation is not there, then you will see deviations from normal gait. This is the boy I showed you on the left side. You see that there are a lot of problems. That means that energy efficiency is not met. We do not know at this point where they are. We know that there are a lot of patients with other neurological pathologies that use aids to walk, that are walking much slower, that costs much more energy, but we know that there's something wrong with the energy conservation. It's not the most efficient pattern, but at this point we cannot say where on the continuum it will be, and it's probably the case that some patients will be very efficient. Other patients with the same syndrome will need to use a lot of energy to walk the same distance. And we're talking about the cost of walking. But those are all things we can analyze and we can evaluate. And that's important because that means that we can look at a patient-specific level what goes wrong. There are so many things in gait that can go wrong. We need much more than just a video to observe. That's what I'm going to talk about in the second part. How can we evaluate gait? Important to know is that if you want to know what goes wrong during walking, you need to know what is wrong with the body. For example, if I see that the pelvis is high on one side, low on the other side, that might be because there's a leg length discrepancy. If I know that there's a leg length discrepancy in stance, I can almost certain say that there will be problems during walking. So every evaluation of gait that we do in a gait laboratory always consists of a thorough clinical examination of the body, where we look at muscle length, or their muscles that are short, or at muscle strength, or their muscles that are weak, but also at leg length, body height, and bony deformities. For example, when we see that children are walking with their foot outwards, that can be because there's a problem with the femur, with the thigh that is rotated more. Of course, if we see it on gait analysis, but we do not know if it happens already before on the skeletal part, then it's difficult to interpret the data. So every gait analysis always consists of a clinical examination. To do this examination, it takes about half an hour. I'm talking about an extensive gait evaluation. If we want to do that, it takes some time, but we can do it in parts. So we do it a lot. We did it in about 50 children with Dravet syndrome, also with intellectual disabilities, and it was a problem in maybe 1 or 2 children who were not cooperative. But it can be done. We take our time. We cannot rush it because often in a clinical situation, a patient has to come back after a year or after an operation. So we never rush a gait analysis because we want to do it again. So we do it at the pace of the child. But it takes some time. It's important to know as a parent that it's an evaluation that takes time. It's not coming in and going out 5 minutes later. The evaluation is done on a table. We measure. So it's nothing that is scary, but some children already don't like to lie on a table, and sometimes we have to do it on the floor. But we always try to find a solution that works for the patient. What we can then do is look at video. A video evaluation. I showed you before pictures. Now I can show you some videos. And then you can see, maybe someone recognizes the child, then you can see that there is something wrong in the gait. But that goes so fast that if I want to evaluate it, I have to stop the video to look at it. I have to make a video from the other side, which I did not do now because of privacy reasons. But it's very easy for you to see that something is wrong. But in order to know what is wrong, and this is easy because he's walking very slowly. That gives us more time to look at it. With some software, we can pause the video. We can make the child walk slower so we can look at it. But still, it's 2 limbs, different joints at the same time that I have to observe. It's very difficult for me, even if I have video evaluation. It's better if we can do it with a standard form, and then we look at each phase of the gait cycle, and we look what is happening there and what is happening there. But it takes the same time it takes me to evaluate the child in the gate lab, I need again to analyze the data. So it takes time, but it can all be done. When I teach this instrumented gait analysis in class, I teach it at the university in the second year of physiotherapy, then all I get is, it's too difficult. But 2 years later they say, oh, we need the graphs because it's too difficult to evaluate the gait without the graphs. So it takes some time to learn the language. But once you know how to speak the language, how to read the graphs, you always want the graphs if you need to evaluate a gait pattern. What can we do? We can collect spatiotemporal data. We can collect movement registration, so what I can see, but we can also collect force data. And force data is something you cannot see, the kinetic data. And we can also look at muscle activation patterns. And those are all pieces of a puzzle, together with the clinical examination, that can then guide me how to treat the child in the best possible way. When you see this, it's a bit what they do with the sporters. When you are in the Vuelta or the Tour de France, they have a whole team that are looking at all the things, and they try to have the footballer or the wielrenner, how do you say it in English? They want to have them at their best level. That's what we try to do. We want to have them at the best level, the best possible level with the deviations. And that's why we need gait analysis. That's why we need to look in depth, because then we can find out how can we gain the other 10% that are really making a difference. That's why we want to look at all those data. Collecting spatiotemporal data in movement registrations is not that difficult, because I can make a video and the child is just walking up and down. If I want to collect force registration and kinetics, it becomes a bit more difficult. Sometimes it's not possible in children who are not cooperative. But if we have already the first part, it learns us a lot. How do we do such a gait analysis? We need preparation time. Almost as much time to prepare the child as that we need to collect the data. Because we're going to put markers on the children. Those are reflective markers, and we're measuring with cameras that are infrared. So they're sending a light, and they're capturing the reflection of the markers. And based on the movement of those markers, we can make a model. We know the height of the child. We know the leg length. So we can make a patient-specific model. Based on that model, we can calculate the angles during walking. It takes some data processing, but no worries, we do that for you. And then we get the first data, the spatiotemporal data. These are the data of one of the children. You see the left side and the right side, and in this column I plotted normal. Now I can look, is there a symmetry between left and right leg? For example, when I look at left and right, the walking speed is the same for the left and the right leg. The cadence is the same for the left and the right leg, and also the step width. But when I look at step length, I see that there is a discrepancy. The step length of the left step is longer than the step length of the right leg. I can then compare those data with normal gait, and then I know something about the deviations, and then I can see that the cadence in this boy is normal, but the walking speed is much less than what I would expect. Normally we walk at 1.1, 1.2 meter per second. It means about 4 to 5 kilometers per hour. If you want to cross a red light, you need to be able to walk 0.8 meter per second. Otherwise, you will not make it. So that's a reference. This boy will need twice the time to cross a red light. So that's really a gait deviation that has a functional impact. If you walk 0.9, you can still go safely into traffic. So that's no problem. It's a slight deviation, but not a real problem. But here you can see that. Step width. Here you see parameters that are lower. Step width we see is increased in this gait. You're still with me after a nice lunch? Okay. Let's look at some kinematic data. Kinematics are describing the movement. I showed you a lot of pictures, but it was very difficult to look at all the moments at the same time. Now I can plot my data from 0 to 100% of a gait cycle. These lines are representing toe-off, that means the end of stance phase and the beginning of swing phase. And now I can look, this same picture is only the initial contact. What goes wrong here? I have a blue line for the right side, a red line for the left side, and I have in gray what should be done in a normal situation. So it makes it very easy to look where there are problems in this gait pattern. Those were the other pictures. Those are just other moments in gait cycle. These are the graphs my students get at an exam. And then I ask, please give me what goes wrong for the hip in the sagittal plane. Then they have to find first the hip. But once you know it, it's easy because everything for the hip is here, plotted here. Then they have to know the plane. That means, how am I looking at my patient? So they just pick, when they know that question, they just pick one graph in the first column, the graph of the hip, and then they can answer the question very easily. It's a lot of data, but it helps us very well to evaluate gait and to make a treatment plan and to see what we can do and change about gait. This is a child with cerebral palsy, and this is then in the transverse plane when you're looking from above. Something I cannot do as a clinician when a child comes into my room. I can ask them walk forward and back, but I cannot go on top to look what is happening. So for rotation deviations, we really like this transversal kinematic data. 2 children. Here we can see the hip flexion extension, what I'm doing with my hip. This is flexion extension. Do I do extension? This is the 0 line. Like I'm standing now, I should be at this line. No flexion, no extension. When I do this, I go to extension. When I do this, I go to flexion. I start normally in flexion. That's the beginning of my gait cycle. But when I progress, I need to do an extension. You see here that this is not happening at all. That means that for the step length, I don't have the extension. I cannot make progression. I can see it very clearly here on these graphs. What is happening during swing? I also see that the knee is not fully extended, and I also see that there are problems with the initial contact of the foot. Same graphs for the other girl, for the girl in this situation. We see that there's also a problem of extension that we did not see on the video, that's clear, but now we can see that it's substantial, but it's not as large as we see it here. We can see that left and right side, in this situation, the red side is doing less. In this situation, the blue side is doing more flexion, so more deviation, so less good. This is the better graph for the blue side. This is the better graph for the red side. And we can also see when it's okay. There are some problems, but it's small problems, and it's mainly in the knee and the hip, and the ankle is doing very well. I showed you a few graphs, but we look at all 12 graphs, of course. Then we can also look at kinetic analysis. And kinetic analysis is very difficult because kinetics you cannot see. That's moments and powers. A moment you can explain when there's a door and they put the handle always on the outside of the door, because then I don't need a lot of force to open the door. When they would put the handle on this side, near to the rotation center, I would need a lot of extra force to open the same door. That's what we're doing. We're creating moments in our joints, and that gives us movement. If we want to look at those moments, we can see here normal moments in gray again. And here we see that this child has an extension moment, where here there's a zero moment. So he found a way that he does not need the muscles. The moment means which muscle group is overactive. There's, in both situations, too much flexion that we saw in the movement description, but now we can see what is happening behind that flexion. So it helps us. Here you see it again in a normal situation. This is excessive flexion. This is excessive extension, hyperextension. And you see that the moment here change the direction. Both are pathologic, but now we can see in which direction they go. And that helps us because then we know which muscles we must address. What we can also do is look if a muscle works, yes or no. And that's an electromyography. We all already saw an EKG and an EEG this morning. Other each, electromyography. We're now looking at the electric activity of the muscles. We look if a muscle is on or off. Like the light, is it on or off? And then the bars, the black bars, they show me where in a normal situation a muscle should work. And then here I can see, okay, the muscle does not work enough, or there is hyperactivity of the muscle. These are now electromyographies of children in cerebral palsy, where there's a lot of spastic muscle movement, so movement that should not be there. We have not looked at the data in children with ADMP syndromes, but we know a little bit from data in children with Dravet that it is difficult to control the muscles. Every time when you need muscle control to move fluently, we see that it's more difficult. So we think if we are going to look at those EMGs, that they will help us learn what are the more difficult parts during walking. Frank promised you a little bit scientific output. What we did already now, a couple of years ago, it was pre-corona, we tested already 7 children. We did data collection and we did analysis of the individual data, so on an individual level that leads to treatment suggestions, advice. We have now, and if you're interested, we can test your child too. We have now 4 days reserved specially for this population that we can test at the motion lab in the USA here in Antwerp. And we have students that are going to analyze the data on a group level, and they're going to compare with similar genetic syndromes, being Dravet syndrome, with Professor Keulemans, who is retired now, and Anne-Sophie Schonjans, who is going to move on with his work in Dravet. That's a different gene. And now also with Anna Janssen, who is looking at the syndrome TSC, which is also both genetic syndromes with intellectual disabilities. And we think that the patterns we see are going to be very similar. But we don't know yet, but what we see, what we observe, we think that there's going to be a problem with flexion, that there are a bit more inflection, but also with the rotations. And I looked already a bit to your children during lunch, and I also already saw that there are a lot of children moving the foot outward. So that's what we think we can see. And the analysis is going to be done in spring because they want their master thesis by the end of May 2023, so we hope we can give you some data there. If you want to look it up, you can also go to this paper. It's a paper we published also pre-corona on the clinical usefulness, but also the challenges of instrumented gait analysis in patients with intellectual disabilities, because it's known for about 20 years in children with cerebral palsy, spina bifida. And a lot of people told us it's not possible to do it in children with intellectual disabilities. We followed 50 patients for 5 years, and I think we had 2 dropouts, but that's all of it. And one of the dropouts now comes for clinical evaluation. So it's not because it cannot be done. It's a challenge, but we like challenges, and it can be done. And if you see how much information it gives you, it's really worth the while to try it. So in Belgium, it's just your physician that has to ask to do this evaluation, and once a year you can have this evaluation on a clinical level. But we also share, of course, if you agree, we also share the clinical data with the science so that we can move on for science faster too. I promised you some treatment options. That's the very last part of my presentation. We heard this morning that there's not only problems with walking, but also problems with motor development in general. Motor development you can see as a mountain you have to climb, and in the beginning, the first weeks, an infant is really moving based on reflexes, but then they are learning postural control, they are learning a lot of movement, and when we go to the fundamental motor problems, motor patterns, we say that is reached when a child is walking independently. And from then on, you go to the top, and here the skillfulness starts about the age of 11, 12 years, but it ends when you go to the Olympics or somewhere, so you can go as high as you want. You don't have to climb the top, and we see that the top should not be the same for everyone, and not for every motor pattern. So, for example, the top for walking can be higher than the top for running. You don't have to reach everything, and there are a lot of, for example, if you don't want to cycle, you don't have to learn to cycle or swim. What can we do in this mountain that your children are climbing? We can help. There are physiotherapists that can help in motor development, that have the right exercises to know if there's a problem with walking or with running, how we can get you maybe not to the top, but a little bit higher than you are now. So that's something we can do. Because motor learning goes by trial and error. And toddlers are learning by trial and error. They fall, they stand up, they do it again. But children with movement problems, they stop after the error. And that's where we can do a lot with physiotherapy in general, stimulating motor development in physiotherapy practice, but also by educating the parents which exercises can be good to stimulate walking, for example. Because to walk, we talked about stability. Children who never crawled don't have a lot of stability in the pelvis. And if the stability in the pelvis is lacking, it's very difficult to walk. So if you can learn them how to get that stability, then walking will be much easier. Guided by clinical evaluation and gait analysis, we know what to stimulate. We know if there are muscles that are short that we need to stretch. We know which muscles are weak that we can train. And we can exercise the difficult movements. Always based on the individual problems. If physiotherapy is not working, there are other options. For example, orthotics can help stretching. Or orthotics can also help walking. When we see children with orthotics and they are prescribed, we always see them again next year to evaluate if the orthosis is doing what she should do. Because when you prescribe an orthosis that is not doing what it should do, then it's a burden for the child. And sometimes we think that might help because we see in the gait there are problems with prepositioning, there are problems with stability. We know that an orthosis can help, but then still we need to evaluate that in the child in the specific situation, because if you see an orthosis that's not working, it's just a burden and a cost, and we should not continue. It's an option, we can try, but not every option works for every child. And that's where gait analysis helps us to find the right option for the problems of specific children. Thanks a lot to the children that allowed, and the parents that allowed us to show the data in this presentation. And I learned this morning that you always have questions or suggestions, so bring on.
questions? Yes. Maybe.
Do you also do a follow-up?
Yes, normally in a clinical situation, we do a follow-up after 1 year. It depends if the physician asks us. But because your child was already in the first study, but you can come again if you want to see.
Interesting for you?
Yes, yes. We did not start really with a large scientific project yet, and I'm looking to it because I'm invited here. But we are planning on comparing them with other intellectual disabilities, and also, of course, the evolution is really important to know. But the first step is to do the cross-sectional examination and to gather the data. But then in the Dravet population, we did a follow-up and we're looking at the longitudinal evolution. But he is welcome to come back, and then he would be the first with 2 analyses. Yeah.
Do we come back on the dates that you just shown us, or?
Those days are reserved for this population, but we can test on any day. But we have a waiting list of 3 months already. But those days, Anke already told us we want to test because it's also part of scientific research. And then we blocked out 4 days for scientific research. But if it's a clinical question, we just can see the patient in the clinical gait lab. So there are going to be more dates in January, but those days are now blocked for you guys.
Okay. I see, Anke, you have something you want to add?
I want just to say that send me an email if you are interested. Some of you have already come, but please contact me, ask me several questions, and we look and see if you could join. So very welcome.
But we can find other possibilities if it's not suiting the date, but those days we blocked because we knew we wanted to do something for the research.
Okay, some more questions? Yes?
I have not a question, but I can only add that my daughter's having this orthotics, but we haven't had this gait analysis, and it is very new topic for me and very interesting. And thank you for bringing this today. And I will write an email to Anke to participate. Thanks.
I'm really because I saw the orthotics in your son. I saw it and I had this feeling it's not doing what they should do. Because the orthotics we know from other pathologies, and if we want to try them in a new pathology, it's imperative that we evaluate if they're doing what they should do. Because otherwise it's a burden and it makes gait less efficient.
I was going to ask, is this a study done only for Belgium patients or also from surrounding countries?
Yeah, I think so. Yeah. Yeah. For Belgian patients, it's paid back by the health insurance, but it's not that there are going to be 100 patients, so we can do it based on a scientific research project. We can see
yeah, and
a few
here already some
patients
out of the Netherlands were included as well.
If it's scientific, it's exactly the same evaluation, but the payment is different. And we cannot, for scientific research, we need money. It's as simple as that. So if there's money, we can see as many patients as we want. But now this year we have students, so we don't need the money because we can ask the students to do it as part of their education. So this year we can see also patients. Now is the moment to see patients from other countries too. Yeah.
Okay, Jesper.
Well, just out of curiosity, my son has a slight scoliosis, and I know a few other ADNP children has scoliosis. Do you have any thoughts on that relating to this?
What we're doing now, in the first analysis we did not, but we are now included the trunk, so that we also see what the trunk is doing during walking. But if there's a scoliosis in stance, it will be there during walking. That's what we know. So it's something we should treat, but it's not always a part of the gait problem.
Are you seeing that these children are having these issues because of contractures? Are you seeing that these children are having these problems because of contractures?
I would think when they walk like that they might develop, but I cannot comment on patients with contractures. But if you're never extending your hip, then you're going to develop a hip extension contractures. Or in the knee, when you're walking in a crouched gait, then we see that if you want to passively extend the knee, that after a while it's not possible anymore. So that's something we should look for. When we see this gait pattern, we always are checking if the contractures develop.
Okay, so I think no more questions. Thank you, Patricia.
Thanks so much. At the same time, I'm a little bit confused because it's my honor to announce Professor Ilana Gozes at the same time, but then a little bit later it's going to be the Zoom. So would you mind splitting your contribution? So start now for 15 minutes and then continue afterwards. I don't know else how to solve it. I'm sorry. Yeah, that's very kind of you. So maybe you can do the introduction here and then because the Zoom is
set. Okay.
Yeah, it's been set. Sorry.
Okay, so actually this takes out a little bit of my anxiety. So I've been been giving lectures. I don't know, I'm not that young, so I've been giving lectures for decades. And every time I give a lecture, I'm very excited. So, and especially I've been told to speak to people, because I speak science. So science has a different language. And I forget that I'm speaking to people, so it's good. It took out my anxiety, so thank you. We have discovered the ADNP gene. I wanted to say the VAP gene, that's the gene that regulates ADNP, many years ago. And we were actually looking for proteins that are protecting the brain. And that's how we cloned it, and we called it activity-dependent neuroprotective protein because it protects the brain against blockade of electrical activity. And without electrical activity, our brain won't work. And in order to develop perhaps a therapy to ADNP, to the ADNP syndrome or Helsmoortel syndrome, we actually need a company because, as you know, clinical development is not cheap, and also the regulatory authorities all over the world, they require very strict regulation. So hence, we started a new company, and I'm going to give you... Actually, can I close the door, please?
But then it becomes too hot.
Okay, because some people are talking outside, and I have a very good hearing. So we started a new company, and we, together with Tel Aviv University, and myself, and also Jeff Schwartz, who is sitting here in the audience, he is the CEO, and Joe Schiarelli, I hope I'm saying it correctly, who is the CFO, Chief Financial Officer, and also we have a clinician who is extremely knowledgeable about bringing drugs to market, and he already brought some drugs to market, so hopefully, together with him, we will bring it to market, to you. And so we are addressing ADNP, a protein essential for normal brain development. And so I'll start from the beginning of ADNP. So at about 1997, actually, I had a very, very good student, Merav Bassan, and her project was to try and find these proteins that protect the brain, and she discovered actually ADNP, and she wasn't sure it was such a good protein. She came to me and she said, am I going to get a PhD for that? I said, don't worry, you will get a PhD for that. And that's how afterwards my career was building on that. So in 1997, we actually wrote a patent, and we had a patent on ADNP. And in 1999, we published the paper in Journal of Neurochemistry, and it was difficult to publish because it was very new protein, and they said, well, what is she talking about? You know? And then I had another excellent student who is now a rector of a university in Israel. It's called Ariel University. His name was Aber Pinchasov. And I said, we have to make a mouse and understand what is this ADNP that Merav was so worried about doing. And so we went to the NIH, and at that time, just, animal models were just starting, and we tried to knock it out. And then he comes to me with a very long face. I'm not getting any mice. What am I going to do? There's not going to be a PhD thesis. And this guy was very, very hardworking. And then I said, why don't we look through our development and see what is happening? And he looked through our development, and this you can see here. So without ADNP, there is no brain. So what happens is, at the time when the nervous system has to form, it doesn't form. So the neural tube, that is how it's called, doesn't close, and the brain doesn't form. But this is when you don't have ADNP at all. If you have half the content of ADNP, so I told him, let's go and see what happens when you have half the content of ADNP, then the mice are born, and they are okay, but actually what they have is the ADNP syndrome. So they have problems. And so we saw that very early on. Actually, I have to say that Albert Pinchasov got a prize for his paper in brain research. It was the best in the year. He got $2,000. He was very happy, but he was more happy with the discovery because he's a real scientist. And what we saw also, at the time when we identified ADNP, we looked at the sequence and we identified all the, you saw all the things that ADNP is doing, but we also identified a very small fragment that we identified before in another protein, and we knew that it is protective. And we called it originally NAP, and now we are calling it davonetide also. We also called it once CP201. So this is the sequence. And remember the beautiful lecture that Claudio gave you about the building blocks, the amino acids that athletes take. And so these are the amino acids of NAP. So it's 8 amino acids. And ADNP, on the other hand, has 1,102 amino acids. And so actually, Merav cloned the mouse ADNP, and then Rachel Zamostiano cloned the human ADNP. And when we looked at the mouse and the human, they were 90% identical. So meaning that what we see in the mice, and you also saw this very beautifully in what Claudio was telling you, is very similar to what we see in the human, and I'll explain. So we have this NAP, which is protective and gives some replacement therapy to ADNP. And what we also discovered is that ADNP is part of a big consortium of proteins that is called, never mind, there is no test about it and you don't need to remember it, so it doesn't matter what it's called, but this complex of proteins is very important for the expression of the chromatin, for the expression of other genes. And we saw that during embryonic development in the mouse, at the time, ADNP regulated about between 400 and 500 genes. And then when we looked postnatally after the mouse is born, we saw that ADNP is regulating thousands of genes. And what is also interesting, it's not regulating it exactly the same in males and females, and also it depends on what kind of mutation in ADNP one has. So it's not, but the pathways, as you also saw from what Claudio said, but also what we have seen before, there are some fundamental pathways that are shared by the different ADNP mutations. And what is now available are actually computational tools that you can make the structure of ADNP. So I collaborate, I don't know how to do it myself, but I collaborate with a computational scientist who actually made the structure of ADNP. So this is in gold here. So this is the structure of ADNP, the three-dimensional structure. And what we discovered, and this we discovered now in 2022, we discovered that ADNP actually interacts directly with another protein that is associated with another autism syndrome, and that is associated with this protein, SHANK3, and this syndrome is called the Phelan-McDermid syndrome, which is more prevalent than the ADNP syndrome, but not by much. But they also, they appear like together when we look at the prevalence of the different syndromes, and you see here from a protein from a paper in 2016. Here is ADNP, and here is SHANK3, and these two proteins are leaders in causing autism spectrum disorder. And why do these two proteins interact? Because ADNP has this NAP structure, and this NAP structure actually binds to this green domain in SHANK3 that is called SH3 domain. So this is a new discovery. We discovered how NAP actually connect these two, or the davunitide, I should say, these two proteins, and also these two proteins, both, you see this red structure, so both these proteins bind another protein in the cell. So if you remember your biology 101, or maybe even in high school, so they both bind the protein that's called actin. Actin is a very important protein for our synapse, for our connections in the brain, and also for the muscle function. And actually in 1975, I published a paper showing that there is actin in the brain. And this is very important for, as I said, for synapse formation. So without actin, SHANK3 won't work. And apparently also ADNP would need it. And that makes the skeleton of the cell. So the skeleton of the cells is made of actin and tubulin. This is the other one. So now I'm talking science again. And this is the way that they interact. And now when there is a mutation in SHANK3 that would cause autism, and what is interesting, some mutations in SHANK3 would cause autism, others will cause schizophrenia. So there is some connection. But when there is a mutation in SHANK3, you see it's all of a sudden it's oriented differently. See, the actin binding site moved. And then what we showed is that actually by treatment with davunitide, we can actually correct it. Meaning that if we can help the ADNP syndrome population, maybe we could also help SHANK3. And I don't have the slides here because this is like fresh out of the oven, but we are also working with Dravet syndrome that we heard so much about. We heard a little bit about, and we can also help the Dravet syndrome because, mice, because the underlying pathology is similar. And I'll explain a little bit more. So this is just to show you, this is, again, I'm a scientist, so apologies. And I'm originally a biochemist working with proteins. And so, and this is the work of my student Maram Ganaim. And what you see here is that if you look at actin interaction with SHANK3 in the normal situation, this is what you see. Then when the SHANK3 is mutated, all of a sudden there is much more interaction. And when we treat with davunitide, it kind of normalizes it. It brings it down. And this is going together with normalizing in a three-chamber, and I'll just explain. It's normalizing the way the mice recognize each other. So the mice are kind of autistic. They won't go to the mouse. They would prefer a cup, an object rather than a mouse. And if we treat them, they will behave normally. So this is what we see here, the mouse and the cup normalized against the abnormal behavior here. And we normalize anxiety, we normal the repetitive behavior and aberrant social behavior. And this was also very recently published. So we have now in hand a very small fragment of ADNP that we think that we call davunitide, that I should maybe say the bottom line here, because we know it for quite a while. It actually has been in 300 individuals in clinical trials, and it's very safe and well tolerated. And so it can go almost directly to ADNP children, and it's the smallest active site of ADNP. It protects against ADNP deficiency while inhibiting nerve cell death. It is protected by our exclusive patent portfolio, and it fortifies the skeleton of the cell. So I showed you now our very new finding that it fortifies actin, but it also fortifies other structure of the skeleton of the cell, and this is like even the transportation system of the nerve cell, which are called microtubules, and I'll show you that. And these structures actually have been associated with many diseases including autism spectrum disorder, amyotrophic lateral sclerosis, Alzheimer's disease, dementias, and we have shown this for davonetide in animal models and also in human studies, because it had been in patients with amnestic mild cognitive impairment, which is a precursor to Alzheimer's disease, and it showed some protection. And also it had been in clinical trials in schizophrenia and showed protection, and we are thinking about this route as well, but it's like a glove to the hand for the ADNP children. And so, again, I'm a biochemist, so I have to show some structures. And so if you look, these are the microtubules, or if you want, you can call them microtubes. And if these fall down, you know, the nerve cells, we have a nerve cell here that will have a long process that will go to the big toe. And we need it to be in perfect shape, and we need also to transport materials along these long processes. So what happens is, it's not like real tubes, because these are dynamic tubes, and they have in the end what is called microtubules end binding protein. These are the green points here. And if you look at the structure, so this is a key point, and what happens is that davonetide actually has this CYP motif that binds, you see, within the 8 building blocks, the 8 amino acids. So it binds directly, this is a signature that binds directly to the microtubule and binding proteins. And the way it works, it's like an amplifier. So you need the right amount, and actually one protein will enhance the binding of another protein. So you need, so what happens is, if you have a positive in ADNP or an ADNP mutation, you don't have enough, and then you need to fortify it in order to get enough, so that the microtubules will move and actually build up the processes and have the processes functioning, and also make the connection, make the brain plastic. So then the child, and also actually the aging adult, will learn, will protect its brain, learn and remember better. So this is the mechanism. And so just to take it to the human situation, so there was a case, a very unfortunate case in Croatia, that a child died from, not from the ADNP syndrome, but from liver transplantation and sepsis. And then the mother actually donated the brain to us as a consortium, actually. And so what we decided to do is to look, we looked at both gene expression and found that the pathways that are associated are very similar to what we see in the mice. And the other thing that we did is we looked to see if we can see a microtubule kind of pathology. So because, as I said, this is the mechanism. And the microtubule kind of pathology that you can see is the position of a protein that is called tubulin-associated unit, or in short, tau. And tau is a protein that deposits, that binds to through this microtubule end binding protein, actually, it binds to the microtubule shaft. And what we have shown is that davonetide or ADNP actually increase.
Just given a little bit of introduction, he's from the Seaver Center, which is in New York. He's working on a clinical ketamine trial, and they're so kind as to give us some updates on, for him, very early hour. Thank you very much. Please go ahead.
Thanks, Frank. It's good to be here. I'm glad that I could join. I know there was some challenges in trying to get some representatives from the Seaver Center to come in person, but I'm very happy to be here. And I can't see you all, and sometimes I have a tendency to speak too quickly. So if someone will have to kind of raise their hand or shout out and tell me to slow down, especially because I'm going to switch to using some slides now. So far, it's understandable though? Everyone can hear me okay? Okay, good. I see, oh, excellent. That's,
sure,
I see
yeah, and sure.
person.
At the same time, I'm communicating with Anna, who has problems, which that's why I'm sort of.
So Frank, I think that the issue, and Sandra is helping me on the back end, if she were to plug in the meeting ID in the numbers as opposed to using the link that you sent, that's what ended up working for me.
Okay.
So it's the 8AO thing.
Okay.
All right, let's see. It's my slides. Share my screen. You're going to tell me that. Okay. Okay, you see the wrong screen there. That's interesting. You see my notes. What's happening there? Do you see my full screen?
We see the full screen. I think it's you are seeing your own notes, and then we see the presentation, the front of the presentation.
Okay, so I see. I can see your screen. What's on the right side of your screen? It's just blank.
No, it's the ketamine and ADNP syndrome. Your title slide,
Okay,
I believe.
wonderful. All right, so we'll start. So I'm gonna share our very early results. I know there's been a lot of anticipation about getting these published, and we finally have it out there. And I'll sort of just kind of go through our thinking, our design, some of the results, and sort of the take-home points, basically. Of course, this story started a couple of years ago, on my birthday, in fact. This is May 7th. I wasn't born in 2020, but Sandra Simone, the head of the ADNP Kids Research Foundation, and Matt Might, these are both parents. And so it's always nice to see when parents drive drug discovery and development. And that's kind of what's going on here, right? I think, if you look back at the history of sort of idiopathic autism, actually, many of the clinical trials were started based on clinical observations by parents in collaboration with their doctors. So Sandra and Matt approached me and said, hey, we have this collaborator at the University of Alabama who has this very fancy artificial intelligence machine. And when we queried this machine to ask the question, which medicines potentially increase the expression of ADNP, ketamine came up. And of course, ketamine has gotten a lot of notoriety, both as a substance of abuse and a potentially toxic substance, but also as a really powerful antidepressant and a very well-known anesthetic in pediatric anesthesiology. So there's a huge amount of literature now documenting its kind of safety in pediatric anesthesiology, and then a lot of emerging literature suggesting some real kind of promising benefits around psychiatric conditions, including depression. And actually, there's a nasal spray form of ketamine that's been approved for depression. And so these guys approached me. I thought, wow, this is kind of a crazy idea. But then when you start to look into the literature, the artificial intelligence machine kind of picked up on this idea that ketamine might induce ADNP expression on the basis of a few preclinical studies. So the first study here shown on the top suggests that silencing the ADNP protein, essentially, or silencing the genes so you're not producing as much of the protein, actually can increase cancer growth or tumor growth in animal models. And then what happened was adding ketamine to this animal model, increase ADNP expression and reduce the tumor growth. So the idea that's happening is kind of, that's interesting, certainly, that it's being kind of activated through the ADNP pathway. The second study in the middle here shows that low doses of ketamine before kind of full sedative doses that are potentially toxic can actually prevent the toxic effects in brain nerve cells. So that's also really important, and it shows the distinction between using low dose and high dose, and even suggesting that low dose in advance of high dose can be protective. The third study suggests the exact same thing, which is that pre-treatment with low dose ketamine can reduce cell death in ketamine-treated rats in this case. And this was a very high dose. And then also the third study suggested that actually ketamine enhanced the expression of ADNP in brain tissue. So these are all, very preliminary, very kind of early preclinical studies, but they all suggest some promise here. And that frankly was enough. And by the way, many clinical trials have started on the basis of much less, but that was enough to kind of, get our heads going and think, well, this is a medicine that's readily available, it's commercially available, there's a lot of literature about it because it's been used in pediatric anesthesia. So let's at least think about trying to get this studied in ADNP syndrome. At the same time, Alana Gozes' group, and I'm sure Alana is there, I'm quite certain Alana is there. So her group has produced some interesting data suggesting that there's a link between ADNP syndrome and glutamate regulation. Glutamate's really a critical neurotransmitter. It's the main excitatory neurotransmitter in the brain. It plays a really important role in synaptic plasticity, in learning and memory and the ability to learn. And what Alana shows in this study is that in mice, again, this is preclinical, in mice missing a copy of their ADMP gene, there's actually decreased expression of a particular glutamate transporter gene. And that's in the hippocampus, which is the learning memory center of the brain, and also in the cortex. And this is compared to wild type mice. And then also actually compared to mice treated with NAP, which we all know now is a snippet of the ADMP gene. So this is kind of pointing to this relationship with ketamine, rather with glutamate, and that's important because this ketamine actually acts through a glutamate transport mechanism. So ketamine, we know, is what's called an NMDA receptor antagonist, and that it essentially increases glutamate functioning by blocking the nerve cells presynaptically to kind of decrease inhibition through this GABA mechanism over here. So these are GABAergic interneurons that are being blocked, and by blocking an inhibitory neuron, we're actually increasing glutamate in the end of the day. It's also known that glutamate sort of downstream can increase other nerve cell growth factors, and it has a very clear effect on promoting synaptic plasticity. And again, high dose is toxic in animal models, but these high doses these toxic effects may be prevented by pretreatment with low dose ketamine. So it does seem clear that low dose is relatively safe. So again, some evidence based on preclinical models that ketamine increases expression of ADMP, some evidence that ADMP has an interesting relationship with glutamate, and we know that ketamine increases glutamate. So this kind of story came together, and we decided it was a good idea to do this clinical trial. The main aim of clinical trials as they begin is really just to show that the medicine is safe in kids, and in this case, kids with ADMP syndrome. That was our primary goal here, is to show this is a safe medicine, it's well tolerated, and then after that, we wanted to see were there clinical effects, and we only used a single dose. So clinical effects after a single dose are kind of questionable. I will say that in the depression literature, there's lots of evidence that a single dose can have an improvement in depression, but obviously we're not necessarily just treating depression in ADMP syndrome. We were looking at a really wide array of clinical symptoms. We didn't know what to see. Again, we were focused on safety, but we wanted to look at aberrant behavior, things like hyperactivity, sensory reactivity, sleep, language, adaptive function. We wanted to look across a whole range of clinical features because we were really thinking, if this is relatively safe, we want to get some information based on this trial to design a better, larger placebo-controlled trial in the future. We were also interested in exploring the feasibility of electrophysiological markers and using computerized eye tracking to kind of have more objective measures of effect. And then we were obviously also interested in trying to figure out whether ketamine actually did increase expression of the ADMP gene. And I'll talk about both of these, all four of these aims briefly today. So this has thankfully been published now. There's a preprint online. It's not been fully kind of proofed yet. I actually just finished proofing it this week, so it should be available pretty soon, but it is available now in its kind of preprint form. So the way that we designed the study, we screened people over about a month. We had a baseline visit with a lot of various clinical assessments, electrophysiological assessments, a lot of safety testing. We did a single infusion using a single dose in all the kids based on their weight, so basically 0.5 milligram per kilo. The dose was infused intravenously over the course of 40 minutes. And then we did 4 different assessments post-treatment. And our main outcomes really were revolving around day 1 and week 1. We wanted to see what the effect was there, and we wanted to then see, did that effect sustain itself at week 2 and week 4? So this is the design. We enrolled 10 patients like very, very quickly. This whole study, by the way, was finished within a year, which is pretty remarkable. It unfortunately took another year to get the study published, or another 6 or 7 months, but now it's finally out there. So the sample was 10 patients, 3 female, 7 male. Average age was 9.5. I think we had kids basically 6 to 12 was the age range. All these kids were pretty severely intellectually disabled, so the average full-scale IQ was 28. Only 4 had autism, which I think is important, but 7 out of the 10 had ADHD symptoms. We had a variety of different genetic mutations, and then this was their race and ethnicity breakdown. Okay, most important, safety. We found this medicine to be safe in a single dose at this low dose. There were really no adverse events that occurred during the actual infusions, with the exception of one patient who experienced some nausea and vomiting. This patient actually had a history of nausea and vomiting, so it wasn't clear whether it was related to the medicine or not. All side effects were mild to moderate. The most common was kids got a little elated, a little silly, which is something you might expect from ketamine, actually. Some kids got a little bit more aggressive. These are mostly kids who had some aggression at baseline, but there was some more sort of disinhibition or aggression. Fatigue was described at about 40%. There were appetite changes, some anxiety. So this is the whole list of side effects. None of these side effects were problematic in the sense that we had to intervene using any kind of additional medicines. And there were no clinically significant abnormalities in laboratory-based measures, in heart monitoring. I mean, there were certainly no serious adverse events. So we definitely came away from this thinking every medicine has side effects. It's not totally clear, you know, we're very inclusive when we document side effects. So we don't necessarily have to determine whether the side effects were related to the medicine or not related to the medicine. In some cases, you can say it's slightly related. In some cases, you can say it's slightly not related. But this is sort of the totality of the things that were described by parents over the course of the whole 4-week trial. And then in terms of results, so we definitely saw you know, very clear reports of improvement by parents, and you know, based on a number of different parent report measures, including the aberrant behavior checklist, the anxiety, depression, and mood scale, the repetitive behavior scale. These are all parent report measures. And again, mainly focusing on the difference between before treatment and then a week after treatment. We saw, you know, clear evidence of improvement. You know, when you start to test lots of different measures, obviously the more measures you test, the more likely you are going to see some significant changes. So that potentially could be spurious, right? It could just be, you know, error. Here, when you just look at the difference between baseline and after 1 week, everything was moving basically in the same direction, which is towards improvement. And so it did seem significant to us. What also was important was the measures that we were using as a clinician, sort of, you know, talking to parents and trying to determine, you know, based on their experience sort of globally, and then also in terms of specific domains like communication or repetitive behaviors, for example, the clinician's interpretation and impressions, this is the clinician being me, was consistent with what we saw on the caregiver report. So the caregivers are filling out these self-report questionnaires, the clinicians are interviewing the parents and sort of measuring specific domains like communication, repetitive behaviors, and the results from both of those things were aligning, all kind of pointing to, you know, clear evidence of improvement. You know, the most important thing to recognize with small studies like this, especially ones with a single dose, is that everybody knew that the kids were on medicine. So there's an enormous amount of bias, right? So I'm biased as a clinician because I want this to work. The parents are biased as parents because they want it to work. So, you know, often it's the case that in small, what are called open label studies without placebo controls, you see lots of improvement. And that's in fact what we saw, lots of improvement. So I can't ignore it and say, oh, this is just all bias, but we do need to put those caveats in there. So we saw improvements across a number of domains, social avoidance being one of them, repetitive behaviors being another one. The third domain that I thought was really important, where there was a lot of convergence across measures, both caregiver reports and clinician measures, was hyperactivity, which we know, I mean, 7 out of 10 of these kids had ADHD, so hyperactivity is very common in ADMP syndrome. So the whole kind of ADHD domain seemed to improve also, which I thought was important. And then also the other domain that popped out to us as, you know, very prominent in ADMP syndrome, but also improved over the course of this trial, was sensory sensitivities. We know these kids have kind of hyporeactivity, especially to like tactile input, and a lot of sensory seeking kinds of behaviors. And those things really improved over the course of our trial. On the left, you see the sensory profile, where actually higher scores are more typical. On the right, you see a measure that I'll talk about in a minute called the SAND, where lower scores are more typical. More typical meaning better, right? So that's sort of aim 1, aim 2. In terms of aim 3, trying to develop more kind of objective measures, that's been a real challenge for clinical trials in general. Frank, by the way, how much time do I have? I know it's 9 o'clock, and I'll show up a little bit late to my next thing, but do you have something, do you have a hard stop at 9?
No, just please continue. We started like 10 minutes late, so.
Okay, cool. So I'll do another 10
on.
minutes.
Everything is here is a little bit overtime anyhow, so take your time.
Okay. So when you talk to the FDA, they'll tell you that treatment benefit is determined by how a patient survives, feels, or functions as a result of treatment. So if you're going to get a treatment approved, at least by the FDA, you need to show treatment benefit. And there's a lot of different ways to kind of assess benefit. So the typical way is patient report, right? That's not going to be reliable in ADNP syndrome. Caregiver reports and clinician reports may be a little bit more reliable, but still subject to lots of bias. The kind of ideal is to do like an objective test, right? So things that, if you were to put somebody in a brain scanner and it showed clear changes, if those clear changes were corresponding to clinical improvements, the brain scanner is blind to treatment. So that's a nice objective test. Electrophysiology may do some of the same things. Eye tracking might do some of the same thing. The clinical outcome assessments in general in clinical trials and developmental disabilities are really, they're very challenging to develop. So we've been spending a lot of time doing that. We piloted a number of different things in the context of this trial as well that will hopefully be able to apply the knowledge to future studies. I'm not going to talk about this, just out of the interest of time, but I will talk about this. This is a sensory assessment that was developed by our chief psychologist, Paige Seiper. This is actually the first known assessment that combines a clinician-administered observation with a caregiver interview to really kind of dig into sensory reactivity symptoms. So this is sensory hyporeactivity, sensory hyperreactivity, or sensory seeking, and across all three kind of visual, tactile, and auditory domains. We know this measure is appropriate for use in severely affected and minimally verbal populations. And we think it's probably a beneficial tool both for research and clinical purposes. And it's now been studied across a number of different rare genetic disorders, including ADNP syndrome. And a paper just put out last year showed that children with ADNP syndrome show significantly more tactile hyporeactivity and sensory seeking as compared to kids with idiopathic autism, where we don't know any of the genetic changes. And as I said, we already use this measure in the ketamine trial, and we're able to see change. So we think it's sensitive to change over time, which is really, really important. The other measure that we looked at in this trial was computerized eye tracking. And there's a lot of different kinds of paradigms that one can use to try to look at either attention or social attention specifically. So this study used what was called the gap overlap task, which measures engagement and disengagement of visual attention to both social and non-social stimuli. We also measure something called the joint attention task, which looks at sort of your eye gaze at either an object, the kind of target, versus a distractor. And the gap overlap task actually was not so helpful with this particular population. We didn't get enough really reliable data because it was such a small data set. But looking at the joint attention task, we thought maybe this tool showed some promise in terms of being a useful marker of change in measuring social attention with ketamine. So specifically for the joint attention task, we collected data from 9 participants. Only 4, though, really had enough trials at all the time points to be included in the analysis. So this is a very, very small data set, but it's a kind of an exciting tool because it's so objective. So what ended up happening in the results was the proportion of trials where participants looked at the target before the distractor. So again, sort of an attention measure actually increased at both day 1 and week 1. So that's what you would expect to happen. And it didn't increase at week 2 or week 4. So that effect kind of waned, which is again, what you'd sort of expect. and the proportion of time spent looking at these targets also increased. But there was no significant change in the delay between how long it took to look at the target, meaning likely the accuracy of attention improved more than the speed of attention, which is just kind of an interesting finding. Again, very small subset, but shows some promise with this measure, and we're going to continue to pilot this in future studies for sure. And then using EEG, electrophysiology, and using something called the auditory steady state response. This is where a participant hears a series of clicks at different stimulation rates. 40 hertz is sort of gamma frequency, and also 20 hertz we looked at, which is more beta frequency. It's recorded with these nets that you see here on the bottom right. And then you look at sort of the coherence between the trials of clicks, and you're using kind of a time frequency analysis. And the thought is that it's supposed to reflect the synchrony or the communication between nerve cells, okay? And also it's known that ASSR, this particular measure, can suggest a balance between excitation of the brain, which is glutamate driven, and inhibition of the brain, which is GABA driven. And again, the results from this very small subset suggest that this particular measure, the ASR, might be a useful marker of change with ketamine, and we also think that the response on the ASSR is actually correlated in some ways to sensory sensitivities, which is kind of what you'd expect, because again, these are auditory stimuli, clicks. So here we collected data from 10 participants. Only 7 were included in the 40 hertz analysis, and I think 8 were included in the 20 hertz analysis just because after the data cleaning. What we saw was the 40 hertz inter-trial coherence actually increased from baseline to week 1. So that's sort of improved synchrony between nerve cells, and the 20 hertz decreased. The 40 hertz is thought to reflect glutamatergic activity, and in part the 20 hertz is thought to reflect inhibitory activity. So that's kind of exactly what you'd expect. And then there were no differences in subsequent time points, suggesting that, again, you see this kind of marker of early change, marker of early, hopefully, improvement, and then a decrease over time because you're not doing repeated dosing. And the thought was that it was also reflecting increased glutamatergic functioning, which is part of what we think about with ASSR. So to summarize, ADNP syndrome, I think, offers a unique opportunity to study a very specific genetic cause of autism, where we really understand the neuropathology. I think it's, we have a lot more work to do to really carefully kind of phenotype and figure out which are the right targets of interest for clinical trials. We have a lot of work to do in terms of clinical outcome assessments and trying to improve them and make them much more specific to ADNP syndrome. I think in order for trials to be successful in the future, we need to really develop and validate biomarkers. And then this idea that ADNP syndrome can sort of provide a window of insight into autism or neurodevelopmental disorders more broadly is really important. So if you can develop a treatment specifically for ADNP syndrome, it may have relevance to autism more broadly because there will be subsets of people with autism and other neurodevelopmental disorders that either look clinically or electrophysiologically or biologically like ADNP syndrome, in which case the treatments developed for ADNP syndrome could actually also have an effect on a subset of people with autism. And so what are the future directions? I think this is what everyone wants to know. We have designed a placebo-controlled treatment trial using low-dose ketamine based on what we learned from this study. We will likely switch from this intravenous formulation, which is really burdensome, to intramuscular administration, which is quite easy. We will probably do a treatment of 8 doses, with 2 doses a week over 4 weeks. And we will do it in a crossover design, meaning everyone will receive drug and everyone will receive placebo like this. And then we'll be able to compare the effect across these 2 conditions. So it'll be a placebo-controlled randomized study, which is a lot sort of more rigorous than what we've done up until now. Right now we are talking to the FDA about how to balance the burden that families experience in participating with trials. We've asked them, for example, can we have parents learn how to do administration at home so that they don't have to come into the clinic for all their drug administration? And so far they've said no. And we're also talking to other sites about also participating so that not everybody has to go to the same site, and that reduces travel burden. So the hope is that certainly by the end of the year we'll have a defined approved trial, hopefully a funded trial, and we'll be able to move forward the beginning of next year. And this is our team. A lot of really kind of important contributions across the board. Oh gosh, I didn't talk about the expression, but that's a whole nother talk. And let's see. So I think Anna is on the line, and she may also be able to stay for some of the panel discussions. Anna Kostic had a huge input on the design and have been very, very helpful along the way. Michael Breen here, who's also, where's Michael? This is Michael. Michael is actively working on the RNA and the DNA expression studies. The RNA expression studies and the DNA methylation studies. So we have, you know, we don't have results yet, frankly. I mean, we didn't see increased expression. We did not see increased expression in our small sample. We saw way too much variability in the expression of ADMP to really draw any conclusions, but Michael is digging much further into this and inevitably will come up with something interesting. And I think future studies with larger samples will be able to much more definitively answer this question, the critical question of whether ketamine increases the expression of ADMP. And I will end with that.
Thanks so much, Alex. You can't see the audience, but I guess you can hear the applause. Thanks so much.
Sure.
Questions? See one in the back. Can I ask a quick question in between? How did you measure the expression, RNA level or protein level?
I think both, but we'd have to ask Michael. You'll invite us back and Michael will give you the whole rundown. And maybe Anna can answer that question also.
Okay, thanks.
So the trial was restricted to kids between the ages of 6 and 12. So the question is, what was the reason for that? And do you plan to have the same age group in the next trial?
Yeah, so the protocol was designed for 5 to 12, and it just ended up being that we recruited 6 to 12. You know, I think the question about, you know, why do we select such a narrow age range? It's a scientific one. We're trying to reduce heterogeneity. We're trying to keep kids sort of pre-pubertal. You can also design trials 5 to 17, but kids look very different, you know, over that developmental period. I think in all likelihood, this next study will also confine it to 5 to 12. You know, the goal here is to put out a solid, rigorous study, you know, with placebo controls, with repeated dosing that shows this medicine is safe and works. And I think that's different than a registration trial where we're going to apply to the EU or the FDA and say we want an approval specifically for ketamine and ADMP syndrome. We don't actually need that so much because ketamine is so widely available, so cheap, what we think to be safe. We just want to put out a study that gives you kind of clear guidelines about, you know, what will be appropriate dosing, you know, what would you want to monitor, what kinds of things you want to be looking out for in terms of side effects, and what kinds of clinical effects would you expect to see. And then it can start to be used clinically. You know, it doesn't have to just be intramuscular. I mean, there's lots of different ways of delivering ketamine. So I think, yes, we will probably continue to be 5 to 12.
Thanks. More questions? There's a question here from the back. No, from the front.
Yeah, so I was wondering why to choose administering the ketamine intramuscularly and not, for example, intranasally?
Yeah, that's a great question. Yeah, I think if you look at the amount of ketamine that we think becomes bioavailable and goes into the brain, you know, there's a hierarchy essentially where intravenous is perfect, right? Right below that at about 93% is intramuscular. And then when you start to get down to intranasal, it's like 20 to 30%. So I think, you know, I've done a number of different intranasal studies with other kinds of compounds, and it becomes a little questionable how much is actually getting into the brain. And the dosing just becomes less clear. I think from intravenous, sort of extracting the dosing with intravenous to intramuscular is much clearer because it's so much closer in terms of bioavailability. So that's why we chose that.
Thanks so much. If there are no, I'm just out of interest. I guess you planning to recruit in the USA exclusively or not? Your patients in Israel.
I mean, no. I think actually, there's ways of collaborating across sites. I mean, I would love to see as many sites as possible, and then eventually there could be ways to kind of combine data. If we all follow the same protocol, and we all design the study in the same way, essentially, then there's opportunities to combine data, which really increase the power to detect smaller effects. So we've been talking to a site in the UK that I think is interested. And yeah, I think in terms of like the very specific study and the IRB approvals and all that kind of stuff, we'll probably combine sites in the US, but I'm happy to share the protocol once it's decided with anybody. And, again, I think the more data, the better, frankly.
Very clear. If the two more questions, maybe, yeah. Questions are coming up. Two more.
Hi, this is just more of a curiosity, and I was wondering if you take into account the possible additive effect of this neurotropic drug. I mean, there are also past sad examples in which some neurotropic drugs have been shown to be very addictive, and I wanted to know a little bit more about this example in particular, and how do you plan to take this into account? Because for chronic treatment, of course, you have to consider that as well. Thanks.
Yeah, no, absolutely. I think, the FDA had a lot of concerns about that. I think in the context of a clinical trial with 8 doses that are regulated heavily by parents, where we're going to have tamper-proof syringes and so on, we don't expect to see addiction. And I don't expect this to be like a chronic treatment that somebody gets, twice a week indefinitely. So I think we'll have to figure out from here, what the right dosing regimen might be. I mean, do you do this for 4 weeks, twice a week, and then, a booster every 3 months? I mean, these are all questions that are really important. And I think the kind of potential abuse relationship is one that you have to consider in terms of thinking about, treatment clinically in the community in the future. But in a 4-week trial, we're actually not concerned about that.
Thanks. On behalf of the speaker. One last question.
Yes, well, you were talking about sharing protocols. Do you mean that you would like to share the protocols with other universities? So, for example, they can start a protocol in Europe? Is that
absolutely, yeah.
And how
absolutely.
far is there already interest from Europe that you know about?
Yeah. There's a site in the UK that's very interested, and I think already has potential funding. And I think as soon as we have a protocol that's approved by the FDA that we know we're going to be going forward with at least 30 patients here, I think that I'd be happy to share that protocol so that people could also recruit. I mean, I think with rare diseases, you need as many sites and as much data as you possibly can get. So I'm really happy to share it. I just don't want, to me, it would be a shame to have different protocols across different sites that we couldn't eventually combine. I'd like to see, at least some shared measures, and so that the same dosing, so that we can sort of, extract some meaning from combining data.
Yeah, the more the better. Thank you for explaining.
We'll thank Alex once again for getting up late and giving such a very clear presentation.
Thanks, good to be here.
It's very clear. Say hello to Joe from me, and hope
Will
to see
do.
you soon live again, Alex. Goodbye.
Okay, thanks. Bye-bye.
Still have half a talk from Milana too, but maybe we should better have our break now and then the talk afterwards. That's fine. Okay, so we have break. You know where it is now. And then Ilana will continue, and then Sandra. Thanks.
So I'm actually going to show some new results also later. And I wanted to say something else. So I'm a scientist. I was actually the best in class in mathematics. So I'm also I try to be as accurate as I can. So I'll try to be as accurate as I can till the end of my talk. So what we saw, that's when I was stopped, we actually saw pathology in the brain that can be associated with what Anke was showing us at the beginning of the day about the brain atrophy. And this is associated directly with the mechanism of action of ADNP, because ADNP, as I said, interacts with the structure that make the nerve cell structure, the cytoskeleton. So it's like scaffolds or building blocks that actually make, it's our skeleton, like our skeleton. So if our skeleton is disrupted, we will go like that. So what happens in the nerve cells, if there is a problem with the cytoskeleton, then this tau protein falls off these microtubules that make the skeleton, and then the cell can actually start dying or not function correctly. So this usually happens in aging, and in healthy aging, it's not so healthy then, but it happens. But it also happens in many neurodegenerative diseases. It happens in Alzheimer's disease, that's the major disease. It also happened in amyotrophic lateral sclerosis, and we were among the first to show it. It happens in other diseases. And what we showed now, which was kind of an original finding, is that it also happens in the ADNP syndrome. And here I'm showing you the hippocampus, and you see here the brown deposits that deposit on the hippocampus, and this is this tau pathology that is also observed in Alzheimer's disease, and you see it here also in the cerebellum. So the hippocampus is associated with learning and memory. We see it also in the frontal cortex, in the prefrontal cortex, which is also associated with learning and memory, and we see it in the cerebellum, which is associated with movement, and we don't see it in a control. And this is a child about 7 years old, and this is a control about 30 years old, and we don't see it at all. And we are not the only syndrome where it happens. I actually participated in an NIH symposium. NIH is the National Institutes of Health in the United States, the largest scientific institute in the world, and it is found in other syndromes like Dravet syndrome, and we are working also in Dravet syndrome, and we are seeing protection. This is very new, and it is seen in other autistic syndromes. So this is something to keep in mind, that there is a problem, could be a problem in development that is similar to a problem that is seen with aging. And we have maybe drugs or our drug candidate that can maybe protect against it. So this is what I'm showing you here. So as you have seen, we actually have 2 mouse models of ADNP syndrome. We have one mouse model that we have developed before you all came to the world, and we published it first in 2007. And in this mouse model, we saw, and this is a mouse model with one allele, one copy of the ADNP gene, one is missing. So many of the children, if they have a mutation at the very beginning of the gene or at the very end of the protein, then they will be like this old mouse model, quote unquote. But we also made another mouse model to mimic the whole gamut of children. And this other mouse model, and this we did by genome editing, so we edited the mouse to carry the most prevalent mutation in ADNP, and that's in tyrosine 719 in men, and it is in tyrosine 718 in mouse. So this is a new mouse model that we published in Biological Psychiatry, which is a very famous journal, in July 2020, and it was, the editor likes it so much that he wrote an editorial about it, and of course I'm proud of that. And as you see, the mouse has the same deposits of this protein tau that you have seen in the child. This is the normal situation. But when we treat this mouse with davunetide, or this small fragment of ADNP, we clear this. And we know how it does it, because what it does, it actually enhances the binding of tau to to the microtubules, which are so important. And these microtubules then remain intact, and the cell stand straight, like my mother always told me to stand straight. So then the cells are actually alive and well. And what also it does, it actually improves, these microtubules are associated with movement of material, which is called axonal transport. And we know that in our mice, axonal transport is disrupted, and we know that when we give them davunetide, after 1 day, we actually can correct it. So it's a very fast correction, actually. And another fast correction is, this is the visual cortex. And remember, some of the children have visual problems. They have this disconnection between the eye and the brain. So this is the visual cortex, this is the brain. And this can be measured by what is called visual evoked potential. And what you see here that the mice with the mutation, this is the blue, they are deficient. They don't look like the red. You see, this is the movement of the electricity kind of in the brain, and I'm not an electrophysiologist. And you see the blue is very different from the red. And when we treat this, after 4 days of treatment, we correct it, and the blue goes together with the green. So this is in the mouse, but this is something that is measurable in children. And this can be measured in children, and this, of course, in children that have these problems, this can be measured. Of course, also connected with everything else that we saw, EEG, that we saw in the previous lecture. But here we know the mechanism, because I'm showing you the mechanism. We know that this is part of ADNP, and what we are doing is we are bringing the business end of ADNP back, and we are correcting it. So this is something to remember. It's not like a therapy that we did a high throughput screening and we saw that something is doing something. Or we are increasing ADNP, maybe. This is bringing back part of the ADNP. We know that we are bringing it back, and we are seeing this result in a very, in an animal model that is very critical to actually study that. And the other thing that I wanted to show you, and I'm sorry that this is a busy slide, but I never learned how to project myself clearly. So we heard about the gate, and we heard about the gate in a whole half-an-hour lecture. We can do gate also in mice. We actually have like a beam that is a see-through, and we can actually look at the paws of the animal, and we can measure the stride side, the push, how much they push, and we can look at it. And what we saw in both our models, which cover the entire ADNP syndrome, what we see that they have abnormal gait. And I'm showing you here the model with the tyrosine mutation. And the model with the tyrosine mutation, as you may recall from Anke's talk, this is actually what we saw in this paper in 2019. It's the most severe. They are starting to walk like a 3.5, which is later than most of the kids. So what we saw here is also a very severe gait problem. And you see the intensity of the way that the paw touches this beam. And then the swing, so how, I can go, so the swing, remember how she showed it? So what we see here, we see a big difference between males and females. And what we see here, though, is that we see a difference, and then we see a correction. We see a difference, and then we see a correction. So we could correct most of the problems that were happening, and we also did gene expression in parallel, and we saw that the gene expression was also corrected. I wanted you to pay attention also. We saw that the mice didn't grow well, and we saw it by weight, and then when we treated, they actually the treated ones are actually kind of in the middle, but eventually they are closer to the normal, so they are significantly different than the mutated mice. And we also saw slowness in eye opening, which is associated with the facial dysmorphism, and we can correct it by treatment. And we also saw developmental delay that we could correct by the treatment. So we have a whole gamut that we can correct by the treatment. And I'm not showing you here, but in the other model, we also showed many times that we can correct cognition and everyday behavior. So So just to show you the other model, because actually the Professor Kolevson showed it to you in a different way. So and the glutamatergic system was mentioned here. So what you see here is in this model particularly, we measured that, and you see this is again in the hippocampus, an area of learning and memory, is that the glutamatergic, the VGLUT1, so that this is the glutamatergic transmission is dramatically reduced here in the animal that has one copy of ADNP, and then we correct it completely by treating the animal with the vunetide. And this is an MRI from a child, and you see that the development of the hippocampus is slowed down in the ADNP child, and we think that we can maybe accelerate it. So this is a study that we just finished. I mean, it's a small study, but what we did is we subjected the parents of ADNP children to a questionnaire that is actually accepted by the FDA as an endpoint for a clinical trial. And usually these changes take a long time. So we actually looked at 2 children that were originally 6.7 years and then 6.6 years, so about the same age. They actually also look a little bit the same. And we looked at motor skills, socialization, daily living skills, communication, and then a combination of this socialization, daily living skill, and communication. And what you see is over 2 years period, so you see a little movement, and you see improvement in socialization in this child and also in this child. But what you also see is a decrease in daily living skill, kind of in the 2 children. And then in this child, you actually see a decrease in the totality of this. So, and we looked also at 27-year-old patients, and persons. And what you see here is that the result is very, very low, meaning that what we see, that there may be also slow development, I'm sorry to say that, slow development, but also some stunting of the development and maybe accelerated aging. And the other thing that we saw, and also Claudio alluded to that he's seeing it as well, we saw that ADNP regulates one of the major genes associated with healthy aging. It's called FOXO3. So it regulates it, and it is associated with very fundamental mechanism. I don't want to bore you with names, but what we saw, the good news is that when we treated with the vunetide, it actually corrected it. So it makes sense because we are bringing back the protein that is missing. A very small part, but an active site of the protein that is missing. So this is what is known in immunology, in enzymology. You bring the active site. And so in terms of planning for clinical trials, so it is a simple intranasal administration. It had been in 300 individuals with no side effects to write home about, really. The only side effect might have been some irritation in the nose, but this is something that can be taken care of. And also it showed increase in activities of daily living in schizophrenia patients. So this is good news, and also increase in learning and memory in prodromal Alzheimer's disease. And so what we are trying is to initiate a pivotal, I should say, phase 2-3, compound safety, and as I said, the compound safety is proven in pediatric patients like children with established ADNP syndrome, and we will also include diagnostic for ADNP, and the administration is intranasal, and we are going to do a double-blind placebo-control study, because otherwise the results are difficult to interpret. And we are meeting with the FDA soon, and we have already applied, and we hope to start the trial in 2023 with your help, because we have to try it with you together. We have to work with you together, and hopefully we can go fast, because we've been talking about it for a number of years now, and move it forward. We have lots and lots of patents to cover it. And what we also need to remember is that I have a ready-made drug. We need to ready-made drug to go, prepared in GMP, so it's good manufacturing. It's ready to go to people. And we also have from the FDA what is called an orphan drug designation and also what is called a pediatric rare disease designation, meaning that if we, the company and you, are successful, then we can sell it to another company, and this is a big incentive to move forward. So this is because this is a novel drug that has never been, it's never been approved. And we have proven safety and efficacy in other models. And also, we could go to other syndromes and other diseases, but this is the disease that we would like to go after. As I showed you, we do have promising results in Phelan-McDermid syndrome, in Dravet syndrome, and in other indications, and the schizophrenia clinicians are actually at our back because they would like to start also a clinical trial. And in terms of the competitive field, so I put us above. So you heard we have competition from ketamine, but I'm going to tell you something about ketamine, and then there are other drugs that are here. So this is the group, and the CEO is here, Jeff Schwartz and Joe Schiarelli. You know me, this is a nice picture. And we also have a chief medical officer who has 24 years of industry experience and has brought a number of drugs to market. So hopefully with this crew, we can move forward. Now, for further information, here is my email, and you know it. And I also would like to thank the ADNP Kids Research Foundation that many years ago nominated me for a prize for a champion of hope. So this hope has to come to fruition, and I want to do the clinical trial. And this is a meeting that we had in 2019 in California, and that was the last meeting I was away, and that was the last time I was in the United States, so I need to go again.
Any questions for Professor Gauzès? Sure.
Well, I saw that maybe it also could be a benefit for ALS, the drug. Would it be, for me, it would be understandable that you will try it on these people because these people have nothing to lose.
This is a very, very interesting thing that you are saying. And actually, Jeff, sitting just in front of you, came to me and said, let's go and do a trial in ALS, and we are thinking about that. You're right.
Yes, because these people have nothing to lose, so I can understand that.
Yes, we are thinking, you are very, very, very right, and we are thinking about it. And I do have a very, see, but the thing is, again, I am a mouse clinician. So I do have a beautiful, in my opinion, a very nice paper on ALS where we showed that it works in the animals again. Mm-hmm. Yes, so this could be, and they do have, and we have shown that they do have the same kind of pathology, and we can protect against it. You are right. But
that's an excellent point. And Alana's paper, Alana published a great paper showing that a mouse model called the SOD mouse model, using davunitide, reversed the motor deficits, the muscle function deficits that are seen in ALS patients. I actually had a cousin who died at 52 of ALS, and it's a very serious disease. And the current therapies that are now out there are palliative. I mean, they're out there, and they'll help a little bit, but not seriously. And so Alana has also shown that ALS may be a tauopathy, and that means that the microtubules, which provide nutrients to the cell body, are dysfunctional. It's something that has been noticed by anatomists for many years, and nobody ever put two and two together. And so we think that davunitide, by repairing the microtubules, will therefore repair the motor neurons and therefore restore function to the motor neurons in the spinal cord. It's not a cure by any stretch, but it may be an effective therapy. So we're talking about starting a small trial up in Rochester, New York, with Dr. John Fox, who was here yesterday. So we'll see what happens. But davunitide is a very powerful drug that has multiple actions on multiple diseases because it interacts with at least 400 different genes that affect the nervous system.
Wait, Nancy.
on the internet, I saw that duvenetina also was tried by Alzheimer or Parkinson patients.
No, not exactly Parkinson's. So there was a trial in amnestic mild cognitive impairment. So that is in patients that are going to succumb maybe to Alzheimer's disease. This mild cognitive impairment is like a precursor to Alzheimer's disease, and that was done. It was done in 144 patients,
Yes.
and it was very safe.
But
And
it was stopped. I saw.
it wasn't stopped. The trial finished. It was a phase 2, and what was seen is that it improved cognition. So it improved. I should be a little more specific. It improved the short-term memory, and this is what is lost originally in Alzheimer's disease. And the patient was seeing like a picture, and then they saw 4 pictures, and they had to say which of the 4 pictures they have seen after 15 seconds. And this improved it statistically significant, and also 4 weeks after it was so this was a trial. It wasn't. It came to the end, and then it wasn't continued to a phase 3 because the company didn't have exactly the funds to go to a phase 3 trials in Alzheimer's disease, which is extremely expensive.
So it was not.
It was not
because of the
continued.
side
It wasn't.
effects.
It wasn't stopped. Do you know about the trial?
It was.
No, but I know, but many, in general, many trials, even if they're successful, the next phase is one dimension higher in terms of cost.
Okay.
Yeah, so you see the...
There's many drugs on the market that sort of, seem to work, but that the company doesn't.
So I was in the company, actually. No, so I was in the... No. So I was in the company, actually. I started this. So I was in the company. That was a company that was started by me 20 years ago. And at that time, we didn't know about ADNP syndrome. What we did know about was that tauopathy, tau, is actually the major tauopathy is Alzheimer's disease. And I still would love to work on Alzheimer's disease.
Yes.
But in Alzheimer's disease, to do a clinical trial, it cost like $100 million. So this is something that the company didn't have. It's just this, you kind of have to continue with a large company. And the large companies in the Alzheimer's field, unfortunately, are like banging their heads against the wall, not working on tau, but working on the amyloid. So this is a different story.
Okay.
And all the trials are failing, but they are still going after that. So that's...
I had a second question. I also read a paper about the NRP that works together with risperidone.
That's my paper. So good, you're a good reader. So that's again in mice. And we did that so NAP is actually the vunetide, and we did it in mice, and we wanted to make a comparison, and also if they work together. We did it with risperidone, and we also did it with clozapine, another schizophrenia
But it
drug.
can work together with
risperidone. Yes, it can work together with risperidone. We got better results when it was together, and with clozapine, which is another schizophrenia drug that is kind of so-so, but it is given, it actually protected against side effects of clozapine, and it was doing very well. So yes, they can work together, and if the child is treated with risperidone, maybe you can reduce the dose a little bit and add the vunetide. Yes.
And last question. My daughter has atypical white matter lesions. Atypical white matter lesions in the brain. Could you say that is because of the tauopathy?
Possible.
Or
the white matter lesion is more.
Explanation?
It's more the myelin. So so we have. It's like what my father used to call a Zolir band. So it's more the isolating material. So that but if you protect the axons, I mean, if you protect the brain, the gray matter, the white matter will also be protected, in my opinion. So I think that it may also help there. And this is actually, we have a little bit of work there. We didn't work very much on that, but I think that if it protects this, it will protect also the white matter, I hope.
Okay, yeah.
Okay. Just 2 questions. Can you elaborate a little bit on the planned clinical trial? You have on the slide that's a multi-center study. Is it planned to cooperate with a center here in Europe?
Yes.
Too? And can you elaborate a little bit on the funding, because that was hugest problem when we met 3 years ago that
I
there was
know.
no funding, and now there seems to be some
So
funding.
now there is a change in management. You know, I'm not the money person. I am the scientist from Israel. But now there is a change in management. But what we would like to do, because there is a requirement for a large N, we need to have many children. Because if we are doing placebo control, you know, we are doing, so in order to get results that we can all really trust, we need a large group of children. So we would like to be totally global. So we are not going to restrict here and here. Anybody that wants to join is welcome, and we want to work together with you. So this is one thing. In terms of the site selection, you know, the site that we visited together, this is a site that I love, and they are very interested still. So any site that will be interested will be checked. So we have a chief medical officer who did it before, and it will have to be done with all the regulatory correct. And this is, again, not my expertise, but it has to be done properly, and the sites are selected so that it will be easy for the patients, etc. And this. And regarding the funding, hopefully I'm working with guys from Wall Street. Hopefully they will find their way to Wall Street.
Okay, well, that's my question. It stopped because of COVID, then there was no funding.
Yeah.
What do you think now into next year it will work? Because now you're talking also about funding.
So again, the
so
funding.
I'm a little bit confused. Is it going to start or not?
So Jeff.
Because we're all one.
So I suggest that Jeff will
parent
answer.
in this room wants a treatment.
It's an excellent question, and one that the management team wrestles with. We're out raising capital now, and we think we have a very good chance of getting the money needed to run the clinical trial. If we didn't think that, we would not be going through this process. It's not in anyone's interest to waste anybody's time and get false hopes. We have
Well, there are no false hopes. There are only
Ja,
false
Sam.
expectations.
Ja, maar beter. Ja, ik sta te wel. Het is voor mij lastig om dit te
pretty confident that we'll be able to do it.
Nathalie moet weghouden.
Confident enough that we're at the FDA right now with protocol assessments. We have a contract research organization in the country that we're working with.
In America or in Europe?
The CRO is in America.
No, it's global, actually.
But well, that's true. They're in London, excuse me, they're in London and Antwerp.
Yeah.
And the trial will be run worldwide. It'll be a global trial.
But you're asking for.
Protocols right now to get it set up.
But you're asking for FDA approval. Is that also valid in Europe?
No, we're not asking for FDA approval, but we're preparing a briefing document and a protocol synopsis.
Okay.
The FDA's already told us that they're going to give us
Okay.
a date for a response. Once we get the date for response, we'll send in the actual protocol.
Yeah.
Again, 30 days after we get the
Yeah.
okay.
But FDA is for the United States only, or not?
No, it'll be U.S. and Europe.
It's US and Europe. Okay, thanks. I didn't know that.
U.S. and Europe right now.
Okay, yeah, thanks. That has changed since I did it the last trial. Okay, or Anke did actually.
I mean, we're talking to the people that have this kind of
Okay, I think for in the interest of time, we have to
thank
thank Dr. Professor Gozes again.
Of course, and then our next and last speaker of today, it will be Sandra, how most of us already know, I think, because she created this really huge foundation already in the Americas. So, okay.
You want this mic? You know what, I'm so short, I need this right in front of me. Okay, so can we just put that microphone? I'm not coordinated enough. I don't have that many arms, even though most Asian parents have 3 arms, right? We use our feet, we use our hands. Oh yeah, okay. Sorry, everybody. I am very blind. Yeah, I only have a few slides. I'm not scientific. I have no scientific slides. I just have cute ones. So hi, everybody.
Oh, pardon me? You can hear me? Sorry, and I have a horrifically high voice. They can hear me now? Yeah. No, they can hear me now. I've got to hit this and move this, Frank. I'm not that coordinated. So hello, I'm Sandra Sermoni. I am the mother to Tony, and he was diagnosed just a few months after Frank's paper, or cognitive genetics paper, was published. So he's been diagnosed for almost 8 years now. I'm also the founder of the ADP Kids Research Foundation and the International ADP Parent Support Group. And I first want to thank Frank and Kelly and Claudio for putting on such an amazing program. I've had the opportunity to spend the last 3 days in the scientific part of it, and it's phenomenal. It's been very impressive and very exciting. I've really been excited to meet everybody here too, so I know I haven't got to talk to a lot of you all, but I'm really excited. Hopefully later we can all chat. So let me just go to my next slide. I am going to quickly try to cover some of the foundation's projects so everyone understands what we're doing, and talk about the importance of building our international ADP community. Similar to the work being done here, we have begun working on mice and want to thank Frank for depositing his mouse into a repository that allows others to work on it freely and easily. And also thank Claudio for being our ADP mouse man because he's doing some really important work that will validate the mouse brain, which we have to get right to test drugs. We refer to the Kooy mouse as the Kooy, his mouse as the Kooy mouse. And we also have funded a creation of a second additional mouse with the second most prevalent mutation through Joe Buxbaum in the US, who works with Alex Kovazon that you just saw, and we refer to that as the Buxbaum mouse. Somehow I envisioned their faces on these little mice. I'm going to make a cartoon. It's the Kooy mouse and the Buxbaum mouse.
I'm not sure I want to see that.
And so our team at the Mind Institute has just started their characterizing of the Kooy mouse, and we'll be doing some different types of translational characterization that I think will work very well with all the work that Claudio's doing. So it's exciting that we're working on the same thing but complementing each other. We've also begun the creation of additional iPSC cell lines. There are several people here who have their own cell lines and fibroblast lines, and we're really hoping to create a sort of an asset or a catalog where we can identify where all the cells are, who has the fibroblasts, so that we can make it easier for other researchers coming around or not duplicating any work and wasting money. We are also working on a blood and fibroblast repository, and we plan to create a catalog, like I said, of all these assets and make these samples available easily to any team who wants to investigate new findings or treatments for ADP with no restrictions. We're also setting ourselves up to be attractive for biotech companies and drug companies who may have compounds or small molecules that they would like to test on ADP, so that we are specifically ready to go. So building a strong foundation of pre-competitive assets is very important, and scientific collaboration is very important because, as an example, I learned yesterday by being here from a researcher right up there that things like hair, which some of us have collected, would be really important to collect them every year to create a natural history study of that product, so that he can look at the natural history of the molecular level. So these kinds of collaborations are really important because we learn things that we as parents can easily do. This is something that we all can do. And so that's exciting to me, and that's kind of the way I work. We've shifted over the last few years to working on sponsored research projects now that we've been able to generate more funding. And we are just searching for meaningful treatments without undue stalls. We all have children, and the time is ticking, and it's 8 years now, and we've had nothing. So we have several projects going on, some of which are similar to the work here in Europe, and that's exciting. And it's also important to be able to replicate information in animal and cell models mimicking ADMP, so we all know we have something valuable to use. We have our Sievers team working on ketamine, as you've all heard. They are also working on their mouse, as well as some drug screening, low throughput screening at a company called Rumi Therapeutics. We have our team that we've just connected with at UC Davis and the Mine Institute, where they are working at investigating gene therapy. That project has just begun. It is a massive project proposed over the course of 3 years, and we hope to very soon have some exciting information from them. We have a team at a company called Rarebase in California working on a small molecule drug screening in combination with artificial intelligence. As you've heard, we found ketamine using artificial intelligence, and we are now grouping with 15 other rare disease organizations to be able to afford something like high throughput screening, where we all put in $150,000 and spent $2.5 million, and we now had all 15 of our rare diseases have the ability to run the entire FDA approved drug library, and we will next move into the non-approved. But luckily, ADMP had the most hits out of all 15, and we have found some really incredible viable drugs that seem, you know, as Alana said, they're just high throughput screenings, but they cross the blood-brain barrier. They are approved for pediatrics. Some of your children I know are on it, not in the dosing and what we need to be on it. So it's we have the opportunity to stumble across other things if we look. This is just a project that I'm really excited about because it yielded really fantastic results quickly, and that's what we're about, is trying to find, you know, therapies quickly. And again, when we talk about screenings of small molecules, just so you know, these are databases of existing potential drugs that could treat ADMP, we would apply to ADMP cells or ADMP animals to see if they could be potentially therapeutic. So we don't just throw them at our kids. Just the only difference between this one is that it's not a narcotic. So there's a lot of concerns about ketamine being a narcotic, and I will explain that later. But these don't have animal model papers like we found for ketamine. So we will need to create those, hopefully utilizing Claudio and some of the other individuals we've met here today. And so basically, we are just not putting our eggs in one basket. We are realizing that we need to take many shots before we hit a goal. And if we aren't shooting at many shots, we're not going to hit a goal. I mean, we need to hit a goal. And I think working together, we can really speed up science. You know, you kind of heard here and there the little things that I've done, and it's things that everyone here can do. We can all work together as parents and really provide information. We can provide hair, we can provide, you know, going and doing gait trainings, and all of that is very important. And that's it for our sponsored research. And now I'm going to go into the next topic, which is information. It is the one thing that I've heard almost every researcher working on ADMP over the last 2 days. Even Alana just talked about her natural history study of 3 patients. We have 436 patients. We can create a natural history study very quickly. We, as parents, want to know more. The researchers won't be able to figure this out unless we add to their knowledge. An example, Anke's presentation was about 78 children. We have over 400 children. Yesterday, I heard there were no missense mutations. We have, or one. Yeah. I mean, we have this information, you need to have it, you know? So, and we need to provide it.
Don't come out with the massive screening studies.
Right, and,
automatically,
yeah.
that's what I told. So
yeah, so.
I also told you they must be there. But
yes, and we as parents can expedite this information. And, you know, like I said, we can provide that, but we just have to all work together and collaborate. And that's one of the reasons why I'm here, is because we're in the US, you guys are here, and we don't get to speak very often, and we don't get to connect, and we don't realize what each other is doing. So it's really important. Real-world data for natural history studies, I know it hasn't been talked about much, are two of the most important things that we can do for a rare disease. Over the 8-year span since this syndrome was discovered, there is not one dedicated global patient registry for only ADMP syndrome. And the beauty is, when we provide these as parents, we get the input. We want to know about medications. We can do surveys on medications. It provides information for us that's helpful, and it provides information for them, because they're the ones who tell me what they want, and then I get it for them. And so the foundation has invested last year into an ADMP-specific, IRB-approved patient registry program, which will allow the teams in Belgium, in Italy, in the US, in Spain, in the UK, all these other organizations who have approached me, they will be able to use all this de-identified information immediately, just as I can share it with her program, or I don't know what it's called. What is it called? The program you were talking about? Or Anke, I'm sorry. Yeah, so this kind of information. Yeah, right. But it's IRB approved. Because it's IRB approved, we can share that information with them, and they can use it. They can't use my 145 children. They can't use that thing that you just saw of mine a minute ago, because I've collected that as a parent, and it's not IRB approved. So we've really worked hard at making it so that we can share everything, because sharing has been a serious problem, and it's halted ADMP progress moving forward for treatments. And our patient registry complies with all European and American privacy laws. It is a small platform used by numerous other similar rare diseases, so it's been vetted. We have begun with only validated surveys and standard to neurodevelopmental disorders, and we will collect the data and work with our researchers both in the US and in Europe, identifying what additional information is needed. But we have a really great start. We just need everybody to get on and participate. And I know as parents we are incredibly busy, and I have to apologize in advance. The majority of the patient registry is very easy, but the first validated survey, and we're using validated surveys because they're what the FDA will allow to use in a phase 3 drug trial, is a very large genomic one. It has a lot of questions, and you will never have to answer them again. Just I'm putting that out there. The rest of them are very short surveys, and we have compiled it so that, like the developmental surveys for the kids who can't walk, you're 6 months old, they're not walking yet, we have it, those coming back to them every 6 months, versus the 14-year-old, like my son, he'll get one of those every 2 years. So we've found a way to also do a natural history study in a way that's very important, where we're getting more information from the young kids, because they're going to provide us the fastest natural history study, but also monitoring all the old kids. So this is not only a patient registry, it will be a natural history study and a longevity study. And we are going to attach it to all of our biobanks and things like this idea of having a progression longitudinal study of material.